Okay, welcome everybody to Module 12 on your experiment dealing with respiration. In this experiment, you'll describe the process of respiration and explain the properties of tissues that are ideal for respiration. In addition, you'll ID the similarities and differences between organisms that respire through their skin, through their gills, and organisms that respire through their lungs. Finally, you'll discuss respiratory terms that relate to lungs, such as tidal volume, minute ventilation, forced vital capacity, and residual volume, and measure human respiratory values during rest and exercise. Upon completion of this lab, you should be able to define respiration, describe its process, explain the properties of tissues that are ideal for respiration, ID the similarities and differences between organisms that respire through their skin, their gills, and their lungs, discuss respiratory terms that relate to the lungs such as tidal volume, minute ventilation, forced vital capacity, and residual volume, measure human respiratory values during rest and exercise, and calculate tidal volume and forced vital capacity. The due dates and deliverables are listed here. Remember to refer to the Canvas due dates and deliverables so that you submit your material on time and are able to get full credit. Materials you'll need include, from your end, clear tape, a pen or pencil, a scale for measuring body weight, a flat item such as a textbook for measuring height, a stopwatch or timer. From the kit, you'll need balloons and a cloth tape measure. You'll also need access to a computer to upload digital camera images and basic photo editing software such as MS Word or PowerPoint to add labs, leader lines, or text to digital photos, and a subject-specific textbook or appropriate reference resource from the lecture content or other suggested resources. Hello. This is our second to last module. In this module, we'll be dealing with respiration. Respiration at its core is the process of taking air into the lungs. A significant percentage of that air, about 20%, is oxygen. The oxygen diffuses across the delicate tissues of the lungs and into the bloodstream, while at the same time carbon dioxide, a waste gas that's the result of metabolic processes that extract energy from the chemicals that we eat, is eliminated. This has to go on continuously because without oxygen, the cells and tissues in our body wouldn't be able to make sufficient energy to sustain life. And if we didn't eliminate the waste gas carbon dioxide, we'd create toxic conditions around the cells and tissues that would end up destroying them. So, if you look on page 3, the materials required that you will supply include clear tape, a pen or pencil, a scale for measuring body weight, but that is optional, a flat item such as a textbook for measuring height, I have here a clipboard, which is just fine, and a stopwatch or timer. And in the kit, what you want to do is go to your S bag here and extract the balloons, okay? I have a couple of balloons in the S bag. Take them out and show them to you. There's one. And there's another one. Okay. You'll also need a computer to upload digital camera images and photo editing software such as Microsoft Word or PowerPoint to add labels, leader lines, or text to digital photos. And of course the material from the lecture side of the course to give you background on what it is we're studying this week in lab. So what is respiration? Well, it's the act by which organisms exchange gases between their external environment and their cells. Respiration is different than cellular respiration. Cellular respiration is the production of energy from cellular processes. Every organism must undergo respiration in order to utilize energy for cellular respiration. The exchange of gases through respiration occurs by diffusion, which is a process that involves particle movement from an area of high to low concentration. For a gas, that relates to pressure. The movement from high to low pressure is essentially a measurement of movement due to concentration. Gas concentrations in a solution, be it gaseous or liquid, are expressed as partial pressures. The amount of a gas that dissolves into or out of a liquid depends on both the solubility of the gas in the liquid and the partial pressure. Gases diffuse not only between the environment and the cells of an organism, but also between two cells within an organism. So what we're talking about with respiration in terms of the most important gas in the air that we breathe is, of course, oxygen. Okay? So oxygen makes up about 20% of air that we breathe on average. Okay? And we require oxygen to make large amounts of ATP in cells. Remember that in the presence of oxygen, we can make 36 to 38 ATP per glucose, but without oxygen, we make approximately 2 ATP per glucose. So you can see why the act of ventilating the lungs, getting air in and out, is critical ultimately to getting oxygen to the cells. What we're going to find out is that complex organisms require oxygen in order to survive. A vertebrate undergoes the following 3 steps during respiration: ventilation, external respiration, and internal respiration. However, less complex organisms do not undergo all steps of respiration. Ventilation is defined as the process of breathing, inhaling, and exhaling air between the lungs and the environment. Only organisms that possess lungs experience ventilation. External respiration is the exchange of gases between the external environment and the blood of an organism. External respiration occurs because of the differences between the partial pressures of gases in the external air and the blood vessels. In order for an organism to use external respiration, the epithelial tissue where the gas is exchanged in the organs such as the lungs, skin, or gills must be moist, thin, and have a large surface area in relation to the organism. Internal respiration is the exchange of gases between the blood and the systemic tissues, as shown here in this figure. So the idea is that in the figure that we're looking at, the lungs serve as the site of gas exchange between the tissues of the body and the air in the atmosphere. And again, the critical gas that we're after is oxygen. So when we breathe in, we fill the lungs with air, 20% of which is oxygen, and that concentration of oxygen is higher than in the blood that's coming to the lungs from the heart. The result is that the oxygen will diffuse down its concentration gradient into the blood that perfuses the lungs. The result is that the blood will become oxygenated. There's a special molecule in red blood cells called hemoglobin, which is very good at binding to and transporting oxygen around the body. You've got millions of hemoglobin molecules in every red blood cell, and you've got thousands of red blood cells per milliliter of blood. And so what happens is, as the oxygen loads into the hemoglobin, the blood increases its oxygen content. It'll turn from a dark to a bright red. It'll then return to the heart oxygenated, and then the heart will pump it to the rest of the body. When this oxygen-rich blood arrives at the body tissues, what will happen is that we will drop off oxygen to the tissues that need it in order to engage in cellular respiration, which is the production of ATP from fuel using oxygen to facilitate the process. And we'll at the same time remove the waste gas carbon dioxide, which is the low energy end product of oxidative metabolism.
That carbon dioxide will be eliminated when the oxygen-poor blood gets to the lungs at the same time as the blood that's coming to the lungs from the heart is picking up oxygen. Okay, and you can see that diagrammed in the right inset here in the figure. Okay, so the act of external respiration facilitates internal respiration, which in turn facilitates cellular respiration. Remember that without oxygen around, we can't really make enough ATP to run the body, and we would die relatively quickly. Now, there are organisms that can survive without oxygen. They're called anaerobes, but they don't do very much, and they usually are relatively simple organisms. For something as complex as the human body or the bodies of organisms that are relatively high on the evolutionary ladder, we require oxygen in order to grow, repair, and interact with our environment because it allows us to make sufficient energy to do so. Some organisms have the ability to undergo respiration through their cutaneous epithelia, which is their skin. This is a very simple way for an organism to exchange gases with the environment, but the outside tissue must be well maintained in order for the organism to survive. One organism that respires this way is the earthworm. The earthworm secretes mucus that keeps its cutaneous epithelium moist. In addition, an earthworm's capillaries run close to the surface of its skin, so the exchange between the blood and the air occurs across a thin region of tissue. Finally, the earthworm has ridges called parapodia that increase the surface area on the outside of the earthworm. Note the characteristics on the earthworm's epithelial tissue shown in this figure that are beneficial for external respiration. And so what we should note is a relatively large surface area compared to the total body volume for this organism, capillaries that run close to the surface, an epithelial tissue that is moist to facilitate gas exchange, and also reasonably thin so that the oxygen in the air can pass through these tissues and into the blood at the same time that the carbon dioxide moves in the opposite direction. Some organisms, such as fish, get oxygen through water instead of air. Fish still need to exchange oxygen and CO2 to survive, but it's more difficult to obtain oxygen from the water than the air. In fact, a fish expends a quarter of its energy for respiration, whereas terrestrial and land-dwelling animals use only about 2% of their total energy expenditure for respiration. Gills are specialized organs that allow the fish to respire. The gills are simply an extension of the pharynx, separating the oxygen from the surrounding water. Gills are moist, thin, and filamentous, and present a large surface area due to their many folds and subsections, all features that make gills favorable for respiration in water, as shown in the figure. Okay. Now again, what do we notice? A moist surface, a large surface area, a relatively thin layer through which the oxygen has to pass, and as a result we have an efficient respiratory membrane. Note, as in the earthworm, the fish also has to eliminate carbon dioxide as it picks up oxygen in order to generate enough energy to allow the body to function. As complex organisms, humans utilize all three steps of respiration: ventilation, external respiration, and internal respiration. The process begins when air moves into the human body through the nose and mouth, then to the pharynx, which is your throat, then to the larynx or vocal cords, and then to the trachea, then to the right and left main bronchi. From there we branch into the bronchioles, and then finally we end up at the alveoli, which is the site of most gas exchange in the human body. You can see in the figure this branching tracheobronchial tree, which we refer to as the conducting division of the respiratory system, is essentially a pipe to get the air down to the regions of the lung tissue where gas exchange happens most effectively. Okay? The alveoli are perfect for this task because they're extremely thin, their surface area is very large, they have an ample blood supply, and they are moist. and the result then is that we can effectively load oxygen into the blood while at the same time removing carbon dioxide. As the air moves through the conducting division, it is warmed and humidified by the moist tissues as it moves through the conducting division's passageways on its way ultimately to the lungs. If any foreign particles enter the airway, they stick to the mucus in the trachea. The ciliated cells beat the mucus and the particles upward towards the esophagus, where they can either be spit out or swallowed. At any rate, they don't end up in the delicate lung tissue, where they could cause irritation, inflammation, and scarring. Once the air reaches the bronchi, the air moves into smaller passages of the bronchi known as bronchioles. These end at sac-like structures called alveoli, as shown in the figure. When air enters the alveoli, the air is 100% humidified, so the alveoli are moist. In addition, the membranes of the alveoli are very thin and have cells of the capillary beds that cover the alveoli, which are also very thin. The oxygen and carbon dioxide only has to cross two layers of cells, one layer of alveolar cells and the other of capillary cells, in order to exchange gas between the air and the blood. Because of this thin barrier, diffusion of the gases occurs relatively easily. The alveoli are circular in shape, which dramatically increases their surface area. Because the partial pressure of oxygen is lower in the blood than in the air in the lungs, the oxygen readily moves into the blood. On the flip side, because the partial pressure of CO2 is much higher in the blood than in the air of the lungs, the carbon dioxide moves out of the blood and into the air and will then be exhaled. So you can see in this diagram how we have the perfect machinery to engage in gas exchange. We have large amount of surface area, we have a moist membrane, we have a thin membrane, and we also have an ample blood supply. So the result now is very easy for us to pick up oxygen from the air we inhale and eliminate carbon dioxide from the blood. We have to eliminate the waste gas carbon dioxide, because if we're allowed to accumulate in the tissue fluids and in the blood plasma, it will cause a reduction in pH that would create conditions that are lethal to living cells and tissues. After the blood in the lungs gains oxygen and loses carbon dioxide, the blood flows to the heart, where it circulates to the rest of the body. The oxygen-rich blood flows through arteries, arterioles, and finally to the capillaries. The cells in the blood contain higher levels of oxygen than the cells of the body, so the oxygen diffuses into the cells where it's needed. Since only one layer of epithelial cells lines these vessels, the oxygen easily diffuses out of the blood and into the surrounding tissues. The oxygen then enters the cells of the body. Carbon dioxide, the waste gas, diffuses out of the cells and into the blood. From the capillaries, the carbon dioxide-rich blood flows out into the capillaries to the venules, and the veins carry the blood back to the heart and pump it to the lungs. You can see this in Figure 7, shown here. After exchange occurs in the lungs, the carbon dioxide may then be expired out of the body and into the air. And so this cycle continues to repeat. Measurement of the volume of gas exchange while at rest is called tidal volume. The amount of air respired can be calculated by measuring the tidal volume and the average number of breaths taken per minute. The amount of air respired per minute can also be called minute ventilation, which is defined as the respiratory rate times the tidal volume. The measure of the maximum volume of air breathed in and out is called forced vital capacity. From these values obtained through forced expiratory volume measurements, predictions can be made for the maximum volume of the air the lungs can hold. There's always some air that remains in the lungs, or the lungs will collapse. This residual air is called residual volume. The residual volume value has to be added in after finding the forced vital capacity in order to measure the maximum volume of the lungs, which is what we call the total lung capacity, or TLC. During exercise, there is an increased need to get more oxygen to the cells of the body and release more carbon dioxide from the body because of the increase in metabolic rate. Minute ventilation increases during light to moderate exercise, mainly due to higher tidal volume. At higher intensities, the increase in the frequency of breathing also intensifies. So what you're looking at in this figure is a spirometer, which is one of the ways that we can measure tidal volume, vital capacity, inspiratory reserve, and expiratory reserve volume. And this serves as a way to determine the efficiency of your pulmonary system. It essentially is a way to assess the strength of the muscles of breathing and the elasticity of the lungs and the condition of the thoracic cavity. In this exercise, you're going to use a balloon to estimate the amount of air breathed at rest and after exercise, and then you'll estimate your lung capacity. You can use a volunteer for this exercise to make it a little bit easier so that you can count your breathing. If you don't have a partner, you can use yourself. Best special note here, a precaution: if you do not perform physical activity on a regular basis, or if you're concerned about performing physical activity, talk to your doctor before doing this portion of the lab. If you're not cleared for physical activity, discuss other options with me, such as finding another individual who is able to do the activity. So the first thing we're going to do is measure respiratory values, tidal volume, and minute ventilation at rest and during exercise. So our first exercise is tidal volume. Special note: do not perform any strenuous physical activity at least an hour prior to beginning this exercise. For the first step, sit quietly and use a timer to measure 30 seconds and count the number of times you breathe in 30 seconds. One complete cycle of inhaling and exhaling is one breath. Next, record the number of breaths in Data Table 1 of the Lab Report Assistant under Trial 1 next to number of breaths in 30 seconds. Then repeat steps 1 and 2 more times for trials 2 and 3. Record the number of breaths taken during each 30-second period under trial 2 and 3 of Data Table 1. Calculate the average of the 3 trials and record them in Data Table 1. Then multiply the average by 2 to calculate the average number of breaths per minute. Record the average number of breaths per minute in Data Table 1. So the idea here is to get some handle on how much air you move in and out at rest when you're doing normal breathing, and to get some idea what that rate is, okay? And that will give you your minute respiratory volume at rest. So we can do an example calculation here to kind of help you out. So let's do this in blue. So let's say that your tidal volume that you measured was 500 ml. Okay, and let's say that you took 60 breaths per minute. Okay, so what would your minute respiratory volume be? Well, you'd multiply these two together, okay, and what you would end up with is 0.5 liters times 60 breaths per minute and this would be liters per breath, okay, and that would end up with 30 liters per minute. Okay, notice that the breaths will cancel, okay, and the units you'll be left with is liters per minute, and since this is a rate, that makes sense. Okay. For tidal volume, you next want to stretch a balloon several times and blow it up once or twice to near its full vital capacity, or near its full capacity rather, without breaking it in order to make it flexible before beginning the next part of the experiment. This is shown here in the figure. Take your regular breath and exhale normally into the balloon. Note, you'll have to use some force to blow air into the balloon near the end of the breath to stretch the balloon in order to expel the full normal amount. Then, after breathing the normal air amount into the balloon, squeeze the balloon neck Twist the neck several times to prevent air from leaking, and hold it tightly closed so that the air does not escape. While still holding onto the neck of the balloon with one hand, hold the end of the flexible measuring tape with the centimeter side up and wrap the tape around the balloon at the balloon's widest point as shown in figure 10. As the balloon is egg-shaped, you will need to apply slight pressure on a flat surface to get the balloon to form a sphere. Use the measuring tape to measure the balloon's circumference as shown in figure 10. And then the next thing we want to do is compute the volume of this sphere. Okay? Release the neck of the balloon so that the air leaves it. Then record this length in data table 2 of your lab report assistant as the circumference of the balloon for trial 1. Repeat steps 6 through 10 2 more times and record each of the values in trials 2 and 3. Then calculate and record the average circumference of the 3 trials under average of trials in data table 2. Then calculate the average radius of the balloon by dividing the average circumference from the average of trials by 6.28, which is approximately equal to 2 times the number pi. You can see this in equation 1. Record this value in data table 3 of your lab report assistant next to average radius at rest. Then calculate the tidal volume at rest, which is the average volume of the balloon after one breath, using equation 2. Record this value in data table 3 by tidal volume at rest in milliliters. Multiply the tidal volume at rest, the average that you computed, by the average number of breaths you take per minute, and divide the product by 1,000 to determine the tidal volume per minute in liters. This is your minute ventilation. Record this value in data table 3 by minute ventilation at rest, and that will be in units of liters per minute. Okay, so what's happening here? You're using the balloon as a way to determine your tidal volume. Okay, the idea here is that the balloon, once you exhale a normal breath into it, will contain the volume of air that you exhale when you breathe in and out at rest. This is without exerting yourself or using extensive physical activity. You want to form that full balloon into a sphere, measure the circumference, use that to determine the radius, and then once you have the radius, you can compute the volume. Okay, and that will be your TV. Okay, so we can indicate here with equation 2 that this is going to be your tidal volume. Okay, your tidal volume. Okay, and your respiratory rate was what you computed earlier using the 3 trials and the timer. So to compute your minute ventilation, you're going to have your tidal volume multiplied by your ventilation rate. Okay. So tidal volume is going to be in liters per breath. Okay. And ventilation rate is going to be in breaths per minute. So the breaths will cancel and you'll be left with liters per minute as your minute ventilation. Okay. Now, very important to note, all right, that the units that we're recording the circumference in are going to be centimeters, which means that this volume is going to end up being in milliliters. Okay. If you want liters, okay, you have to remember that there's 1,000 milliliters in 1 liter. Okay, so if you ended up, say, with a tidal volume of, let's say, 500 ml, and you wanted to know the number of liters that would be, you would multiply that by 1,000 ml on the bottom and 1 liter on the top. Milliliters would cancel, and you would end up with 0.5 liters. Okay? So that's how to work out the math.
If we begin with the experimental protocol on page 11, okay, it's entitled Exercise 1, Measuring Respiratory Values, Tidal Volume, and Minute Ventilation at Rest and During Exercise. And we have a note up here. A volunteer is suggested in order to help you count your breathing. If no partner is available, try and do this yourself. And also note, if you do not perform physical activity on a regular basis, or if you are concerned about performing physical activity, consult a doctor before completing this portion of the lab. If you're not cleared for physical activity, discuss other options with your instructor, such as finding another individual who is able to complete this activity. So, essentially, if you're not cleared for exercise, try and find a partner. So first on tap on page 11 is tidal volume. Tidal volume is defined as the amount of air that you can move in and out of the lungs with normal relaxed breathing. So again, cautionary note, do not perform any strenuous physical activity at least 1 hour prior to starting this exercise, because that'll change the data that you collect. So sit quietly using a stopwatch or a timer to measure 30 seconds and count the number of times you breathe in 30 seconds. One complete cycle of inhaling and exhaling is considered to be 1 breath. Then record the number of breaths in data table 1 of your lab report assistant under trial 1 next to number of breaths in 30 seconds. Repeat steps 1 and 2 2 more times for trials 2 and 3. Record the number of breaths taken during each 30-second period under trial 2 and trial 3 of data table 1. And then step 4, calculate the average of the 3 trials recorded in data table 1, then multiply the average by 2 to calculate the average number of breaths per minute. Record the average number of breaths per minute in data table 1. Then stretch up one of the balloons several times and blow it up once or twice to near its full capacity without breaking it in order to make it flexible before beginning the next part of the experiment. And the way to do that is shown in figure 9. Okay, figure 9 is at the top of page 12 in your lab protocol. Okay, so again, we begin with the 30-second tidal volume. Okay, so I've got my timer. Okay. I go ahead and push, and then we're going to go 30 seconds, and I will count. Okay, so that was 12 breaths in 30 seconds. And then you would repeat that trial 2 more times and enter the data in data table 1. Okay. Now let's go ahead and stretch and inflate the balloon as indicated in step 5 so that we're ready for our other volumes. So stretch this sucker out and then blow it till it's almost completely full up. Okay, I'm going to go ahead and do the next 2 30-second trials. You don't have to watch me do it since you've seen it already performed 1 time. And then I'll join you for the next step of the protocol. Okay, I've finished my tidal volume trials and I've averaged them. And now we need to get a tidal volume. That's the amount of air that you exhale with a normal resting breath. So we've got our balloon prepped. Okay. You want to take a regular breath, exhale normally into the balloon after breathing the normal amount of air into the balloon, squeeze the balloon neck, twist the neck several times so that the air doesn't leak out, and hold it closed so that the air does not escape. While you've got it closed, you want to measure the circumference of the balloon at its widest point. Now, to do this, you're going to try and get the balloon to be as close to circular as you possibly can. That might require that you press down on one end of the balloon a little bit. And the reason for that is that the formula you're going to use to compute the volume of air in the balloon is for the volume of a sphere. Okay, so we're going to have to use the number pi to compute that. Okay, the measurement technique is shown in Figure 10 at the bottom of page 12. So let's give this a shot. Okay. Now, see how we're a little bit oval here? I'm going to press down on the balloon about like that so that it's round. And you want to measure this using the centimeter side of the tape measure. And what you can do to make it a little bit easier, if you like, is to take a piece of clear tape and tape the zero end of the measuring tape around the belt buckle of your inflated balloon to hold it in place. And then we're going to wrap all the way around. We're going to try and get the circumference of this sphere at its widest point. And what I get is, call it 31 centimeters. Okay? So that's my tidal volume. Now, go ahead, release the measuring tape from the balloon, let the air out, okay? And repeat the process a couple more times to correct for error. And you're going to enter that data into data table 2, which is in the middle of page 17. Okay? So let's try one more time. About 34 centimeters. Okay. Last trial. About 40 centimeters. Okay. You're going to average those. So add those up, divide by 3, and get the average. Okay. And then, once you've done that, you've recorded the data in data table 2. You've recorded the repeated trials in data table 2. Step 13 is to calculate the average radius of the balloon by dividing the average circumference, or the average of the trials, by 2 pi. Now, what is pi? It's an irrational number, meaning it's a non-terminating, non-repeating decimal, 3.141, and then the decimals just go on and on and on. Most calculators have a pi button. You're going to divide your average by 2 pi, or 6.28, which is an approximation of 2 pi, and then record the value in data table 3 of the lab report assistant next to average radius at rest. Then you're going to calculate the tidal volume at rest, which is the average volume of the balloon after one breath, using equation 2. Okay? So equation 2 is the volume equal to 4 pi times the radius cubed over 3. The first equation, which we're using in step 13, and again, this is at the top of page 13, is the circumference divided by 2 pi. Okay, so you want to go ahead and plug those numbers in and get your values. And then in step 15, you're going to multiply the tidal volume at rest by the average number of breaths you take per minute, and then divide this product by 1,000 to determine the tidal volume per minute in liters. Okay, that would be liters per minute that you move in a normal set of breathing cycles over 60 seconds. This is your minute ventilation rate, or V sub E, and then record that value in data table 3 by minute ventilation at rest in liters per minute. Okay, so I'm going to go ahead and run those calculations, and I'll show you basically how they should turn out, and then we'll move on to forced vital capacity. Okay, after compiling my numbers, okay, for tidal volume, I had the following values. Now your values will vary, but this is just to give you an idea of whether or not you've done the calculation properly. I had trial 1, 12 breaths in 30 seconds, trial 2, 13, trial 3, 12, for an average of 12.33, and that would be 24.6 breaths per minute. My balloon circumference was 31, 35, and 40 centimeters for trial 1, 2, and 3 respectively, for an average of 35.3 centimeters circumference. So when I use the formula to calculate the average radius, I get 5.6 centimeters average radius. Tidal volume at rest in milliliters, 745 milliliters. And my minute ventilation, which is the amount of air that I move in and out of the lungs at my resting rate of respiration, in 1 minute came out to be 18.3 liters. Remember that there's 1,000 milliliters in 1 liter. Okay, so when you go to convert your milliliter tidal volumes, when you divide by 1,000, you got to move that decimal place to the left 3 spots.
Okay, the next thing we want to measure is forced vital capacity. In the following steps, you'll estimate your body surface area by measuring your height in centimeters and your weight in kilograms. So first, measure your height in centimeters using the measuring tape. If a volunteer is available, ask them to help you with this measurement. Remember to remove any footwear and stand with your back against the wall, and then use the flat surface of a book to place on the top of your head in a level position. Ask the volunteer to hold the flat surface in place, or you can hold it in place until you can make a small pencil mark just below the flat surface of the book. Then use the tape measure to measure the height of the tape on the wall in centimeters. This is similar to the height measurement that we did in our previous module, Module 11. Okay. Record your height in data table 4 with your lab report assistant. Then use a bathroom scale or similar scale to measure your weight in kilograms. If you don't have a scale available, estimate your weight from a previous body weight measurement and record your weight in data table 4. Use the following formula, the Mosteller formula, to estimate your body surface area, or BSA, and record your body surface area in data table 4. Okay? So remember, we're getting your height in centimeters and your weight in kilograms. Okay? So now you might say, well, wait a minute, my bathroom scale measures pounds. Okay? Well, there's a way to go from pounds to kilograms, and I'll show you how to do that. Okay? Let's say, just for fun, that you weighed, 150 pounds. We just pick a number out of the blue. Okay? And what we want to know is how many kilograms is this? Okay? Well, there's 2.2 pounds in 1 kilogram. So put 2.2 pounds on the bottom down here, and put 1 kilogram on the top. The pounds will cancel, okay? And you'll end up with 150 over 2.2 as the number of kilograms, okay? And if you work that out, that's going to end up being something a little bit less than 75 kilograms, approximately. Okay. Then you want to use the following equation to estimate your forced vital capacity. That's the equation on the bottom. Okay. Your height in centimeters you got from the tape measure. Your weight you can compute by converting pounds to kilograms. You plug that into the formula and you get your BSA. And then you obviously record your sex and you use the appropriate formula. So, for forced vital capacity, you want to take a deep breath, as much as you can possibly inhale, and then quickly exhale fully into the balloon. When you feel that you're finished exhaling, exhale a little bit more. So you really have to blow this out, okay? While still holding onto the neck of the balloon with one hand. Hold the end of the flexible measuring tape with the centimeter side up and wrap the tape around the balloon at the widest point. Again, deform the balloon so that it's spherical. Then record the circumference in data table 4 as you did in trial 1. The balloon circumference should be larger than the data taken for tidal volume, hopefully. Okay. Then repeat steps 21 through 23 from your protocol 2 additional times, recording each trial in data table 4. Record the largest circumference next to the balloon for the 3 trials in data table 4 next to largest circumference of trials. Okay? So in all of these, when you're doing these measurements, you want to make 3 runs and take the average. And the reason for that is to correct for random error. Okay? That's why we do that. Then use equations 1 and 2 to calculate the approximate radius and then the volume of the balloon after exhalation. Record the volume in the column entitled Volume of the Balloon using the largest circumference and FVC estimate from data collected in milliliters. This volume is your forced vital capacity measurement. So if you get the idea here, you're max blowing into the balloon, tying it off, making it spherical, measuring the circumference, and then you're using, again, these 2 formulas to compute the volume in the balloon. Okay, but you're going to get some bigger numbers this time. Okay. All right. Once you've done that, you recorded the circumference in data table 4 as trial 1, and you've repeated this 2 additional times. Record the largest circumference of the balloon for the 3 trials in data table 4 next to largest circumference of trials, and then use that equation to approximate the radius and then get the volume. And then you're going to record this next to volume of the balloon using the largest circumference. This volume is your forced vital capacity. In order to obtain a full lung volume, the residual volume also has to be taken into account. Now, you can't blow out your residual volume or else your entire airway would collapse. So what you do is you convert the units of the FVC estimate from the data collected from milliliters to liters by, again, dividing the number of milliliters by 1,000, just like we did for tidal volume. If you're a female, you want to add 1.1 liters to your forced vital capacity, and if you're male, add 1.2. And this will give you an estimate of the total lung capacity. Okay? And you can again record this in the data table.
Okay. All right. So next on tap is forced vital capacity. This is at the bottom of page 13. In the following steps, you'll estimate your body surface area by measuring your height and your weight. Okay. So you want to measure your height in centimeters using the measuring tape. If a volunteer is available, ask them to help you with this measurement. Remember to remove any footwear and stand with your back against the wall. Use a flat surface of a book to place the top of your head in a level position. Ask a volunteer to hold the flat surface in place, or you can hold it in place until you can make a small pencil mark just below the flat surface of the book. Then use the tape measure to measure the height of the tape of the wall in centimeters. Okay. Now, I already know my weight, okay, and I can convert that relatively quickly to kilograms. Use a bathroom scale to record your weight after you've recorded your height in step 17 and put it in your lab report assistant. Using a bathroom scale, get your weight. You're going to weigh yourself in pounds, and then you're going to have to convert that to kilograms. So there's 2.2 pounds in 1 kilogram. So the way that you would do that is you take your weight in pounds, divide that by 2.2, and that would be your weight in kilograms. Then use the following equation to estimate your forced vital capacity in data table 4 and your body surface area in step 19 using the equation shown there. Okay, so the BSA formula in square meters, that's the amount of flat space your body covers, is your height in centimeters times your weight in kilograms over 3,600. And then for your forced vital capacity, you've got two formulas, one for males and one for females. For females, it's the BSA in meters squared times 2 liters. And for males, it's the BSA in meters squared times 2.5 liters. Okay. Next, take a deep breath as much as you can inhale. Quickly exhale fully into the balloon. When you feel that you're finished exhaling, exhale a little more. While still holding onto the neck of the balloon, hold the end of the flexible measuring tape and wrap the tape around the balloon at the widest point, just like you did for tidal volume, but the balloon will be bigger this time. Record the circumference in data table 4 as trial 1. The balloon circumference should be larger than the data taken for the tidal volume. Repeat these steps from 21 through 23 2 additional times so that you get 3 trials to correct for random variation. And then record the largest circumference of the balloon for the 3 trials in data table 4 next to largest circumference of trials. Okay. Next, step 26, use equations 1 and 2 to calculate the approximate radius when the volume of the balloon is filled with your forced vital capacity. Okay. Record the volume in the column and tile volume of the balloon using the largest circumference and FVC estimate from data collected in milliliters. This volume is your forced vital capacity measurement. In order to contain full lung volume, the residual volume has to be added. You convert the units of forced vital capacity estimate from the data collected from milliliters to liters by dividing by 1,000. If you're a female, add 1.1 liters to the forced vital capacity, and if you're male, add 1.2 liters to your forced vital capacity to get an estimate of the total lung volume. Record the sum in data table 4 by estimated total lung volume in liters. Okay? So I'm going to go ahead and run those numbers for myself, and you can figure out whether you're doing the computation correctly. Okay? You'll see me measure my height, again, using the clipboard here, okay, to record. And the height's going to be in centimeters. And then I'm going to convert my weight to kilograms. I'll plug this into the formula, and you can see essentially what my forced vital capacity is. Okay, so stay tuned. I'll run those numbers for you. I'll demonstrate how to take the height, and then I'll rejoin you for the next installment, which is ventilation measurements during exercise, which begins on page 15. Okay, so I'll see you in just a second. Okay, so what I wanted to do is run real quick through the numbers that I computed for my forced vital capacity, okay, giving you the data I have on myself. All right, this is on page 18. These are my numbers. Your numbers will vary. My height measurement, I'm 5'8", so that works out to 172.7 centimeters. My weight measurement, I'm 255, and that works out to 116 kilograms. And my BSA, using the Mosteller formula, is 2.36 square meters. My forced vital capacity, estimate from body surface area, works out to 5.9 liters. Okay. Now, for my forced vital capacity and estimated total lung volume, I had to take the largest circumference of 3 trials of blowing up the balloon with as much breath as I could possibly exhale. My first trial was 61 centimeter circumference, then 62, then 63. So the largest of those 3 was the last trial at 63. And the volume of the balloon, using the largest circumference, works out to 4,228 mil, which is 4.2 liters. The FVC estimated from the data collected Using the formula in the protocol, worked out to 4.2 liters, and my estimated total lung volume, adding 1.2 liters to the 4.2, gives me 5.4 liters. Okay? So those are my numbers. Your numbers will be different, but I just wanted to give you an example of how to do the calculations so that you know you're doing them properly. Remember to use the calculator and always double check your work.
Okay. Next thing we want to do is measure your ventilation volumes during exercise. So you want to choose an exercise of your choice. Some repeated rhythmic exercise, such as running in place or doing jumping jacks, that you can sustain for 1.5 minutes. Put the balloon within reach and start timing with a stopwatch or timer, and immediately begin to perform the cardiovascular exercise of your choice for 30 seconds. After exercising for 30 seconds, count the number of times you breathe in the following 60 seconds while maintaining your exercise. Count one complete cycle of inhaling and exhaling as one breath. After counting your number of breaths for 60 seconds, pick up the balloon while still maintaining the exercise and use one of your final breaths during exercise to blow the exhalation into the balloon. This is the amount you would normally breathe out during the exercise. Twist the neck of the balloon and hold the balloon closed so you can measure the circumference following the exercise. Then record the number of breaths that you counted in 60 seconds in Data Table 5 of your lab report assistant under Trial 1 next to number of breaths per minute after 30 seconds of initial exercise. Then measure and record the circumference of the balloon using the measuring tape, wrapping the tape around the widest portion of the balloon as you did previously when you did your tidal volume and your forced vital capacity. Record the circumference under Trial 1 next to circumference of the balloon at the end of exercise in Data Table 5. Repeat steps 30 through 36 in your protocol 2 more times and record these values in Data Table 5. These will be Trials 2 and 3. Rest as long as you need between trials. Calculate the average of the 3 trials, then calculate the average radius of the balloon and the average volume of the balloon using the same methods as during resting measurements. Record the average radius of the 3 balloons in the row titled Average Radius of the Balloon in centimeters, exercise measures in Data Table 5. Calculate the volume of air exhaled each breath during exercise using the same methods as during your resting measures. Record the average volume in a row entitled Average Volume of Air Exhaled Each Breath During Exercise in Data Table 5. Calculate the minute ventilation during exercise by multiplying the average volume of air exhaled each breath during exercise by the number of breaths per exercise after 30 minutes of initial exercise. Then record the value in the row entitled Minute Ventilation during exercise in liters per minute in Data Table 5. Okay? And so this will give you an idea of your overall efficiency of pulmonary function. Okay? This gives you something to compare to your age and sex group to determine whether or not the elasticity of the lungs and the strength of the muscles of breathing and the dimensions of your thoracic cavity are within norms for your group. Okay, there are tables that you can use to compare your measurements to average measurements for people in your age and sex. And this will inform you as to whether or not you need either medical advice or perhaps an exercise regimen in order to improve your cardiopulmonary function. Okay, so that brings us to the end of this module. Again, stay tuned for actual demo, and I will see everybody in the final module of the term. I thank you for listening. Okay, we're going to begin the first trial. Jumping jacks is the exercise that I have selected. Okay, and I have a timer that I'm going to use that I've actually set up on my computer. All right, and it'll ding at 30-second intervals. Okay, so when our mark gets set and go.
Okay. That was my last breath. Got the balloon clamped. Now we're going to measure the diameter. Okay, so it's about 61. Record. Okay, so I've got the circumference and then the number of breaths that I took and after the 30-second interval of the exercise. Again, this is one of the reasons why it might be useful to have a partner. Keeping count. So it was about 24 breaths in 60 seconds. Okay. Sorry, that's for 30 seconds, so it should be 48 breaths in 60 seconds. Okay. Then I'm going to enter that data in data table 5 and then run my numbers. Okay. So my first trial was 61 centimeters on the balloon, and then I'll run 2 more trials. I'll give you those data, and then we'll go ahead and compute the circumference of the balloon at the end of the exercise, do the average of both the number of breaths per minute and the circumference of the balloon. And then we'll crank out the numbers. Okay, so I'll join you in the last installment and let you know what those numbers are. Okay, so I've run the numbers, and again, what I have for my height measurement, I'm 5'8", that's 172.72 centimeters. I'm 255, my weight measurement kilograms is 116, and my BSA is 2.36 square meters. So my FVC from the body surface area measurement is 5.9 liters, okay? And then when we did the forced vital capacity for estimated total lung volume, okay, this is again from data table 4, I got 4.2 liters for my FVC, and my estimated total lung volume was 5.42 liters. All right. Now, moving to data table 5, my numbers for trial 1, number of breaths per minute after 30 seconds of exercise, was 48 for trial 1, 52 for trial 2, and 50 for trial 3, for an average of 50. The circumference of the balloon was 61 centimeters for trial 1, 62 for trial 2, and 61 for trial 3, or sorry, 65 for trial 3, for an average of 62.6. The average radius of the balloon in centimeters turned out to be 9.96 centimeters. The average volume of air exhaled with each breath during exercise, again, using the formula from the protocol, 4.14 liters. I took an average of 50 breaths in 60 seconds, okay? And that works out now to a V sub E of 207 liters per minute, okay? So what that shows you, okay, when you compare that to what the resting levels are, a resting VE was 18.37 liters per minute, so it went up almost tenfold, okay? Now, again, this is going to be proportional to your fitness level and your activity level, but why do you think the minute ventilation during exercise increased versus the minute ventilation during rest? And the answer, hopefully, that you'll come up with is that when you're performing some kind of an exercise, the muscles are generating activity, and as a result, they need more oxygen and nutrients. As a result, signals from the brain go to the respiratory muscles and to your heart and end up increasing your heart rate and your respiratory rate so that you can get more oxygen-rich and nutrient-rich blood to the muscles more quickly so that you can sustain the activity. Okay, so that's the entire idea. So hopefully this made sense. Again, make sure to answer the questions at the end of the protocol, and I'll see everybody in the last podcast dealing with microbial growth. Thanks to everybody for listening, and I'll see you in the next module.